So we're going to get started, and I'm going to first talk about mutations and mutants. So bacteria can exchange genes. So bacteria and archaeogenetics and horizontal genes transfer, which is the movement of genes between cells other than reproduction. And there's mutation, which is heritable change in genomes. This can lead to a change in properties of an organism, some beneficial, some detrimental, but most have no effect. Exponential growth in prokaryotes accumulates mutagens quickly. Horizontal genes transfer, which is aka genetic exchange, generates much larger changes. Mutagen and genetic exchange fuel evolution. Continuing on that, genomes of cells are double-stranded DNA, while viral genomes are double or single-stranded DNA or RNA. The wild-type strand is isolated from nature. Wild-type you can also refer to just one gene. So for a mutant, cell or viruses are derived from wild-type that carries a nucleotide sequence or a genotype. And a genotype is designated by 3 lowercase letters followed by a capital that are all italicized. For example, lowercase HIS capital C is involved in histidine biosynthesis. Mutations designated, which are the ones I just said. Observable properties or phenotype, these are like what you can see, may also be altered. So a phenotype is designated by a capital letter and then 2 lowercase letters and then a plus or a minus sign. For example, it would be capital H lowercase is plus sign, which makes histidine. So examples of phenotypical traits include like glucose fermentation, production of enzymes or catalase, etc. This can be obtained either from wild type or parental strain, which is derived from the wild type. More on mutations and mutants, we have isolation of mutants, which is screening versus selection. So selection is a selectable mutations confer an advantage. Under certain environmental conditions, mutated progeny cells outgrow and replace the parent. For an example, antibiotic resistance. They're relatively easy to detect and they're a powerful genetic tool, which can be used in genetic engineering. Screening, however, is a non-selectable mutations do not confer an advantage, even though they may lead to a phenotypic change. For an example, color loss in a pigmented organism. It requires laborious, time-consuming screening, examining large numbers and looking for differences. Blue and white colony system in bioengineering requires screening, which is very time-consuming. Okay, so now we're going to talk about some mutations and mutants. This is isolation of mutants, screening versus selection again. So you have to understand the physiology is key to design the genetic screens. Selection is preferred whenever possible. Common classes of mutants and their detection include auxotroph, pigmentinless, and sugar formation. So auxotroph is the inability to grow on a media lacking selected nutrient, like lacking selective amino acids. Pigmentinless is a loss of enzyme in biosynthetic pathway, results in the presence of different color or lack of color. And sugar fermentation is the loss of enzyme in a degradation pathway, or the lack of color change in a solid medium that has sugar and a pH indicator. So now we're going to start talking about molecular bases of mutation. So the source of these mutations are spontaneous versus induced. So our spontaneous mutations, these occur without intervention. Most are or have occasional errors by a DNA polymerase during replication, versus induced mutations cause an intervention by an environment or by a researcher deliberately on purpose. Exposure to like a natural radiation or chemicals that chemically modify DNA. The amount of nucleotide base pairs affected by the change. So we have point mutations, and these change 1 BP. So change only occurs in one base pair. Occurs via a single base pair substitution. So A instead of G and C instead of T. The effect of substitution, or aka phenotypic change, depends on the exact location, divided into missense, nonsense, and silent. These are all of our different types of mutations. More on that we have on our base pair substitution. So we have missense, nonsense, and silent mutations. But not all mutations change polypeptides due to degeneracy of genetic code. Each amino acid is coded by more than one codon. Wobble hypotheses, for example, which means there's room for error in the third base. Silent mutations do not affect sequence of encoded polypeptide or phenotype. Example, UAC to UAU. A missense mutation changes sequence of amino acids in a polypeptide. For example, UAC to AAC. If not an active site, could alter activity of a resulted protein. And not all missense mutations lead to dysfunctional or non-functional. So we have transitions and transversions. Our transitions are within purines, and our transversions are in pyrimidine to purine, or vice versa. And then finally, we have our nonsense mutations. These are our stop codons, and the chain gets interrupted. So this typically results in truncated or incomplete protein that lacks normal activity, very serious effects. So point mutations that result in an increase or decrease of base pairs. We have frame shifts and other insertions or deletions. In our frame shift mutations, a single base pair deletion or insertion will result in a shift in the reading frame during translation. So the code is triplectic, so each codon that is made of 3 nucleotides corresponds to 1 amino acid. So this will scramble the entire polypeptide sequence downstream because every codon changes. Insertion or deletion of 2 pairs also causes frame shift. Insertion or deletion of 3 pairs adds, deletes a codon and an amino acid, which usually is not as bad. Insertions and deletions can result in a gain or loss of hundreds to thousands of base pairs. These can often result in complete loss of gene function and can be lethal. So now I'm going to talk about reversions and mutation rates. So mutation rates depend on frequency of DNA changes and efficiency of DNA repair. Reversions, or back mutations, and suppressors. So reversion, it occurs because point mutations are typically reversible, whereas a revertant is a strain in which original phenotype is restored, and the mutation somewhere else in the same gene that restores the function. Our mutation rates. For most microorganisms, errors in the DNA replication occur at a frequency of 10 to the negative 6 to 10 to the negative 7th per kb. So eukaryotes have a 10-fold lower error rates than microbes. DNA viruses have error rates 100 to 1,000 times greater. RNA viruses' error rates even higher due to less proofreading and lack of RNA repair mechanisms. Mutation rates in our decreasing order are our DNA virus is greater than our RNA virus is greater than our DNA virus, which is greater than bacteria. Alrighty, and that's all I have to say about my topics.